Dear students, today we will discuss on diseases of mango and its management. Mango is an evergreen tree in the family Anacardiaceae, grown for its edible fruit. The mango tree is erect and branching with a thick trunk and broad rounded canopy. The leaves of the tree are shiny and dark green. There are several factors which hampered its production. This topic will discuss on the detailed discussion of major diseases of mango and its management. So coming to the first disease, that is powdery mildew. Now let's see the symptoms of the disease. Pathogen attacks the inflorescence, leaves, stalk of inflorescence and young fruits with white superficial powdery growth of fungus, resulting in the shedding. The affected flowers fail to open and may fall prematurely. Initially, young fruits are covered entirely by the mildew. Fruits may remain on the tree until they reach up to a marble size, and then they drop prematurely. Infection is noticed on young leaves when their color changes from brown to light green. Next is the reason for severity: high humidity, cloudy weather and high wind velocity for 3 to 4 days. Minimum temperature, that is 10 to 13 degrees centigrade, maximum 27 to 31 degrees centigrade, and relative humidity of 82 to 91% are most conducive for disease severity. Next is the mode of spread. Spread is dependent on development of mildew pathogen persist on older leaves or malformed panicles during off season. Disease spread through wind-borne spores, which takes 5 to 7 hours for infection after germination. Now coming to the management of the disease. Prune diseased leaves and malformed panicles harboring the pathogen to reduce primary inoculum load. Spray wettable sulfur at the rate 0.2% when panicle sizes are 3 to 4 inches. Spray Dinocaf at the rate 0.1% 15 to 20 days after first spray. Spray Tidomorph 0.1% 15 to 20 days after second spray. Spraying at full bloom needs to be avoided. So this is all about the powdery mildew. Next important disease is the anthracnose. It is caused by Coletotrichum gliosporoides. Next is the symptoms of anthracnose. The pathogen causes leaf spot, leaf blight, withered tip, blossom blight and fruit rot. On leaves, characteristic symptoms appear as oval or irregular vinaceous brown to deep brown spots of various sizes scattered all over the leaf surface. Disease also produces elongated black necrotic areas on twigs. The tips of young branches start drying from tip downwards. The blossom small black spots appear on panicles and open flowers, which gradually enlarge and coalesce to cause death of flowers. The infected flowers fall off, leaving more persistent spikes on peduncles. This is all about the symptoms of the disease. Now coming to the mode of spread. Disease spread by rain splash, spores, infect all above healthy tissues of the host. Fungus also produces spores on dead twigs, leaves, inflorescence and fruits, which further make the disease more complex. Free moisture available in the orchard enhance disease spread. Pathogen remained viable for 14 months on fallen leaves, dead stem and diseased twigs attached to trees. Temperature 25 to 30 degrees centigrade with 95% relative humidity for 12 hours is conducive for development of disease. Now coming to the management of the disease. Diseased leaves, twigs, gall meats infected leaves and fruits should be collected and burnt. Blossom infection can be controlled effectively by spraying of Bevestin at the rate 0.1% at 15 days interval for 2 to 3 times. The foliar infection can be controlled by spraying of copper oxychloride at the rate 0.3%. Pre-harvest sprays of hexaconazole 0.1% or carbendazim 0.1% at 15 days interval should be done in such a way that the last spray falls 15 days prior to harvest. Hot water treatment at 52 degrees centigrade plus minus 1 degree centigrade for 20 minutes. The duration can be reduced to 10 minutes by supplementing carbendazim or prochloras both 0.05% in hot water. Covering the fruits on tree 15 days prior to harvest with news or brown paper bags. Use biocontrol agents such as Streptosporangium pseudo-vulgare. This is about the management of the disease. Now coming to the next important disease, that is the dieback. It is caused by Lesiodiplodia theobrome. Symptoms of the disease: the pathogen causing dieback, tip dieback, graft union blight, twig blight, seedling rot, wood stain, stem end rot, black root rot, fruit rot, dry rot, brown rot of panicle, etc. It is characterized by drying black of twigs from top downwards, particularly in older trees, followed by drying of leaves, which gives an appearance of fire scorch. Internal browning in wood tissues is observed when it is slit open along with the long axis. Cracks appear on branches and gum exudes before they die out. Now coming to the reason for severity: lack of proper cultural operation such as pruning of diseased twigs and application of pest around tree trunk. Tree damaged by gummosis, insects, drought and lack of nutrition favor the disease development. High summer temperature predispose the plant to the attack of pathogen by reducing the vitality of the plant. Next is the management of the disease. Scion wood selected for propagation should be free from infection. Every care should be taken to prevent introduction of disease in newly planted orchards. Any infected portions should immediately be pruned, then spray or paste with copper oxychloride at the rate 0.3% or with cow dung at the cut ends. Pruning should be done in such a way that some healthy portion is also removed. to ensure complete eradication of the pathogen. The affected branches should be collected and burned. Next important disease is the sooty mold. Causal organism is Capnodium mangiferae. Now coming to the symptoms of sooty mold. It is very common wherever honeydew secreted insects, that is mango hopper, scales, coccids and mealybugs are found. Black velvety, thin membranous covering on leaves, stem and fruits are the symptoms. Now coming to the management of the disease. If honeydew secreting insects are controlled by suitable insecticides, the mold dies out for want of suitable growth medium. Spraying of monocrotophos at the rate 0.05% 2 to 3 times at 10 to 15 days interval is quite effective. Spraying of starts at the rate 2% is also effective. Spray wettable sulfur with monocrotophos and also gum of acacia at the rate 0.2 plus 0.05 plus 0.3% or Indian oil formulation number 1 and 2 at the rate 4% at 15 days interval. Application of pesticides should cover both sides of the leaf. Now coming to the next disease, that is the Phoma blight. It is caused by Phoma glomerata. Symptoms of the disease. The disease is noticed on matured or old leaves only. Fully developed spots are characterized by dark margin and dull grey necrotic center. In severe cases, spots coalesce to form patches, which results in withering and defoliation of infected leaves. Now coming to the management of the disease. Spray copper oxychloride at the rate 0.3%. Balanced nutrition provides resistance to Phoma blight. Next disease is the bacterial canker. It is caused by Xanthomonas campestris, Pathova, Mangifera indica. Symptoms. The disease is noticed on leaves, leaf stalks, stems, twigs, branches and fruits. Initially producing water-soaked lesions, later turning into typical canker. On leaves, water-soaked irregular satellite to angular raised lesions are formed. On fruits, water-soaked dark brown to black colored lesions are observed, which gradually developed into cankerous, raised or flat spots. These spots often burst, extruding gummy substances containing highly contagious bacterial cells. Now, reason for high severity. Disease development is favored by high humidity of 90%, moderate temperature of 25 to 30 degrees centigrade, and high wind velocity is congenial for severity of disease. Now coming to the management. Regular inspection of orchards, sanitation and seedling certification are recommended as preventive measures. Mango stones for raising seedlings, that is the rootstock, should always be taken from healthy fruits. Use of windbreaks helps in reducing bruising, wounding and thus reduce chances of infection. In severe infection, spraying of streptocycline at the rate 300 ppm or copper oxychloride at the rate 0.3% is more effective. Next is the malformation. It is caused by Fusarium subglutinans. Symptoms of the disease. First is vegetative malformation. Vegetative malformation is produced in young seedlings. The affected seedlings develop vegetative growth which are abnormal growth, swollen and have very short internodes. Next is the floral malformation. The flower buds are transformed into vegetative buds and a large number of small leaves and stems developed and reduced internodes, giving an appearance of witch's broom. The flower buds seldom open and remain dull green. Next is the management of the disease. The floral malformed panicles or vegetative malformed shoots should be pruned and burnt. Application of NAA at the rate 200 ppm or Planofix at the rate 90 ml per 20 liter before bud differentiation stages, followed by deblossoming or bud burst stages, reduce the incidence of malformation. Spray chelated zinc ion at the rate 100 ppm or copper ion at the rate 40 ppm during flower bud differentiation and flowering stage, combined with spray of carbendazim at the rate 0.1% during November-December. Next disease is the gummosis. It is caused by Lasiodiplodia theobrome. Symptoms of the disease. The disease is characterized by the presence of profuse oozing of gum on the surface of the affected wood, bark of the trunk and also on larger branches, but more common on the cracked branches. In severe cases, droplets of gum trickle down on stem, bark, turn dark brown with longitudinal cracks, rots completely, and the tree dries up because of cracking, rotting and girdling effects. Management of the disease. The diseased bark or portion should be removed or cleaned and pasted with Bordeaux paste or copper oxychloride paste or cow dung paste. Application of copper sulfate at the rate 500 gram per tree in soil around the tree trunk is recommended. Next disease is the red rust. It is caused by Cephalourus virescens. Coming to the symptoms of the disease is readily recognized by the presence of rusty red fructification of the algae on the surface of the leaves, petiole and young twigs and fruits. Initially the spots are greenish grey in color and velvety in texture, which finally turn into reddish brown. Now coming to the management of the disease. If vigor of the plant is maintained by balanced nutrients, the disease is less. Spray Bordeaux mixture, that is 5:5:50 or copper oxychloride of 0.3%. As the disease starts on the onset of rain, it is desired to spray fungicide twice during the month of July or August at 15 days intervals. Next is the physiological disorder, that is black tip. It is also called as the chimney disease. Symptoms of the disease. Symptoms become visible when the mango fruits attain some size. Small etiolated area develops near the distal end of the fruit, which gradually spreads, turns nearly black, and covers the tip of the fruit completely. The black area remains hard and the growth of the fruit is checked. Next is the reason for high severity. Through toxic gases, that is sulfur dioxide, ethylene, carbon monoxide and fluoride emitting from brick kilns operating nearby orchard. Now coming to the management of the disease. It can be minimized by the spray of borax at the rate 1% or other alkaline solution like caustic soda and washing soda. The first spray should be done positively at P stage, followed by 2 more sprays at 15 days interval. Planting of mango orchard in north-south direction and 3 km away from brick kilns reduce the incidence. So this is all about the mango diseases and disorders. Now coming to the conclusion of the topic. Mango is affected by many diseases like powdery mildew, former blight, anthracnose, bacterial canker, gummosis, sooty mold, red rust, and also physiological order such as black tip. These diseases are of national importance and causes economic losses in mango. Proper diagnosis, monitoring of mango orchard and timely application of control measures such as application of fungicides can minimize these diseases.